Hello, everybody.
Professor, I had a question about the last exam.
Yes.
Were most of the questions from the exercises in the book?
No, none. None that I know of. Maybe
Oh, because
some were similar, but that was not my intention. Why were there some the same? Have
to
to
be
check.
honest, I studied the notes and the slides, but the questions were like so difficult for me. I don't know if it was for other people as well. Like I totally felt that I didn't know anything. So I was and I don't have access to the book. So maybe I was
No,
thinking, is
they
it
were
from the
not.
book?
No, they were not. They were
Oh,
not from the book. Hmm. Okay. Okay, if there are any more questions about the exam, I'll answer them at the end of the class. We'll go over the questions. Okay. Okay, let's see. Are there any? It looks like they have their own. Okay, so do you see, everybody sees the slides, right? So this is lecture number 18.
Yes, we do.
Good, thank you. I felt the same way. I read the textbook. I think what would help is finding application-based questions online. Any good websites for that? So take notes during class, review them over the next day, and watch the recording as well to make sure it stays in your head. Okay, these are all good suggestions. So we'll talk about the exam at the end, okay? You can't download the lecture because it needs a password. I don't know. That's anyone have that problem? Do you want to download the lecture? I don't think it's password protected. Or maybe it is. Let me
There's somebody saying to check the wait room. They're waiting to get into the lecture.
Oh, thank you. I don't see anyone waiting. Maybe it was a few minutes ago, because right now I let everybody in. Now there's one. Okay. So let's see. Yes, the slides and the lectures should be able to be downloaded. Okay. Oh. Oh. What's going on? Waiting room participants. Oh, there's no one in. Oh, is that what it's showing up? 19. Oh, oh. Okay, let me change this quickly. Sorry. I will change the lecture posting. It's from last year. So sorry. I'll do it right now. Okay, let's go to bright space. Okay, can you tell me if you can download it now, please? It should be okay. Yeah, the notes is one page. Can you imagine? Okay, so now I think we're all set to go. Okay, so you have the slides now, right? Okay, so we're going to start a new set of lectures, which is the nervous system. And it's my favorite system of all, because that's what I work on for many, many years. Okay, so what is here? What I'm showing you is a histological preparation of a neuron from the rat midbrain. So you have to start hearing all these names, because remember that the brain is a map of sensations. All our taste and vision, for example, is located here. All our sensations end up in a specific area of the brain. Each area of the brain has its specific function. And by the end of this set of lectures, hopefully you will have learned all of them. Why can't I see? There's one person in the waiting room. Ugh, so annoying. Not show me the participants. Okay, now I think we're all set. Let's see. So you can see the slides, correct? Yes?
Yes.
Okay, great. So I was saying that this is a neuron from the midbrain. There. Okay, what you see here is the cell body, this sort of triangular area, and then you see these spider-like projections. And you have to think about this as an electrical unit. So these projections are wires that are going to conduct the electrical current that we've been talking about when we talk about muscles. So the action potentials here are going to be conducted along these very thin projections that are known, some are known as dendrites, others are known as axons. So here's one, and this is a neuron from the thalamus. And the thalamus is an area in the brain where all the input that comes from the periphery is going to end up. All the electrical signals that come from the periphery that are initiated by activation of receptors that are on the skin or the eye receptor, the olfactory receptors, and so on, they convert the signal into an electrical current. That electrical current is going to be conducted by these wire-like projections of the neurons. And they're first going to end up in this area that's known as the thalamus. It's an area in the middle of the brain. And from there, then the electrical current is going to be spread out to the different areas of the brain. So it's the brain is an amazing system, very complex, more complex than AI, you can imagine. AI is nothing compared to the brain. Okay, so here I wanted to show you this slide because these are neurons, human neurons that we are growing right now in my lab. So they were derived from a cell taken from an individual, a skin cell, a fibroblast. You know very well what a fibroblast is. So then those fibroblasts were cultured in a plastic dish, and then with a collection, a cocktail of chemicals, they were differentiated. So they went back to their pluripotent state. That means that they were not differentiated. They were no longer fibroblasts. They were just an undifferentiated cell that then, with another cocktail of chemicals, was converted to neurons, but it could have been converted to cardiomyocytes. And that's the beauty. So the investigator that differentiated or that discovered the chemicals, the cocktail of chemicals necessary to convert pluripotent cells into neurons or to differentiate cells, won the Nobel Prize. Okay, so here you see neurons, you see with the projections, some are dendrites, others are axons. Here's the cell body in green. And it's green not because our neurons are green. It's green because we stain the cells with specific chemicals that makes them green. And this is from an individual, a healthy individual that has no Alzheimer's. And you see that the projections are pretty intact. Here on the right, these neurons are from an individual that has Alzheimer's, or neurons similar to an individual that has Alzheimer's. And you see that some of these projections are broken apart so that the electrical current, the action potential, cannot be conducted. So this is just to give you an idea of the complexity and the beauty of these neurons that allow us to perceive changes in the environment outside our body and to generate messages to the different parts of the body, specifically to muscles and glands, to bring the body back to homeostasis. How do we grow? In dishes, and it takes a long time. Look, these neurons have been 28 days in culture. These are my students that are magicians, not me. This is all made by my students, PhD students. Okay, so now I'm going to show you a small video that's going to show neurons in the hippocampus. So the hippocampus is another, so I talked about the thalamus. This will never go back. I talked about the thalamus, and which is the area of the brain where all the outside sensation is going to be transmitted to. The hippocampus is the area that is dedicated to short-term memory. This is the area that is degenerated in Alzheimer's patients. And it's called, what I'm going to show is called the brain bulb. And you see that neurons here are colored. They have all these different colors. And these different colors correspond to neurotransmitters that these neurons carry. So someone in Harvard was very intelligent and creative and was able to devise a technique to color the neurons in the brain with different chemicals so that corresponding to their neurotransmitters, to the different properties of the neurons, so that they would have different colors. And that's why it's called the rainbow, like the rainbow. So you can see here. So here first, these neurons are not colored, but these are neurons in the hippocampus, so they carry memory sensations. So nothing to do. You see here, beautiful neurons, all with the projections, different colors, and they depend on their different properties. Okay, now let's see, how can I get out of here? Ah, there we go. Ah. Okay, so Here's another type of neuron. These are neurons in the myenteric plexus. What is the myenteric plexus? Myenteric plexus is a nervous system that is dedicated solely to the digestive tract. Neurons are in the adult. Trillions, many neurons. And we are trying to understand the organization of the brain, but it's very hard. We start by the brain of a fly or a brain of a worm. C. elegans only has 200 neurons. So it's easier to study. Is procedural memory encoded in the hippocampus as well? So the two areas of the memory is hippocampus and then frontal cortex. So short-term memory, all types of memories are in the hippocampus. When they become long-term, when they become solid memories, such as, for example, riding a bike or something that you learned in childhood that you'll never forget, that's already stored in the frontal cortex. Okay, so these are other types of neurons, and they're the autonomic neurons, those that are not under voluntary control. Okay, so what am I going to show you here now? So what is the main role of the nervous system is to generate a quick response, one of its functions, to maintain homeostasis. Of course, the nervous system is also involved in intellectual ability, intellectual activity. So to maintain homeostasis, we have these two systems, which we already, you know this by heart now. The endocrine system, which you will study in the spring, generates a slow response, involves hormones, and they have to be carried through the blood. That's why it's slow and reaches the target, but it's much longer lasting. The nervous system responds very quick, and it involves these electrical currents that we call action potentials. So that's what we're going to study now until the end of the semester, is the nervous system. So neurons aren't able to regenerate. That is correct, but not 100% correct. Now we have knowledge that perhaps there's some neurons that are stem cells, that are precursors, and that are hidden, for example, in the hippocampus, the memory center. And under certain conditions and in response to certain chemicals and signals, they can maybe generate new neurons. In rodents, for sure, there are some neurons that can be activated. There's some precursor neurons that can be activated and give rise to new neurons. In humans, it's still a little bit controversial. Okay, so what do I want to show here? So here is an axon of a neuron. The cell body would be outside of the screen. Here's the terminal, the synaptic terminal, and here's another neuron. This is the cell body, and these are the dendrites. So what I'm going to show you is how this action potential is going to be transmitted from this neuron to this neuron. So how can we differentiate between these two neurons? You see that the neurons do not come into contact. Just like if you remember, we have a motor neuron. When it reaches the muscle fiber, it's not in physical contact. There's a little space, which we call the synaptic cleft. Here's the same system. The two neurons don't touch physically. So there's a space in between two neurons that are communicating. They don't touch physically. There's a space in between that's called the synaptic cleft. And when an action potential is traveling down the axon of the neuron and reaches the axon terminal, you know that there's neurotransmitters that are being released into the synaptic cleft. These neurotransmitters, here represented by these little dots, will diffuse across the space and bind to receptors on the dendrites or cell bodies or axons of the other neuron. So this neuron we would call presynaptic. This neuron we would call postsynaptic, so that you know, you get used to all these terms. What's the difference between synaptic cleft and synapse? Okay, so this here is a synapse, this whole unit, because it has the axon terminal of the presynaptic neuron, and it has the dendrites of the postsynaptic neuron, and it has the synaptic cleft. So the synaptic cleft is the space. The synapse is the whole unit. Okay, so a synapse is when it's going from neuron to neuron. So synapse is a physical entity. It's a space that's made up by the axon terminal of the presynaptic neuron and the dendrite of the postsynaptic neuron. Right. So synaptic cleft. The synaptic cleft is the space between the two neurons, where the neurotransmitter is going to be released. Okay, now watch the action potential coming down the axon. When it reaches the axon terminal, the neurotransmitter is going to be released, and it's going to trigger a new action potential that's going to go down the postsynaptic neuron. So if there's a signal that is strong enough, there's a change in potential, you see the action potential coming down the presynaptic neuron, then you have the neurotransmitters being released and generating an action potential in the postsynaptic neuron. And this goes on and on and on from neuron to neuron until it reaches its target, until it reaches the final
So I'm writing an essay on.
There we go. Here, good. Okay. So with all this beautiful introduction to neurons, the synapse is the same as a neuromuscular junction. Yes, good, very good. It's a synapse between two neurons, neuromuscular junction between a motor neuron and a muscle. Yes, perfect. Okay, so what are the functions of the nervous system? So here we have three functions: sensory, integrative, and motor. Sensory, what does it mean? It senses changes. It senses changes in the internal environment inside our body and in the external environment, the environment around our body. And how is this possible? Because of receptors. What are receptors? Receptors are proteins that change their shape when they are stimulated, when they are activated, either by a photon, by a sound wave, by touch, by taste, whatever. These receptors will change shape, will change their tertiary structure, quaternary structure, and they will emit a signal, an electrical signal. Okay, so we have receptors inside, receptors for oxygen, for carbon dioxide, for thirst, for pH, and so on. Outside, we have the eyes, the nose, the tongue, the ear, touch buds, and so on. And those are all the perceived changes in the external environment. Okay, so we generate a signal in the receptors. This is the sensory function of the nervous system, and it's transmitted to the central nervous system. The central nervous system is made up of the brain and the spinal cord. So the central nervous system is the integrative part of the brain. It's the control center. So it's made up of the brain and the spinal cord. So this is where these electrical signals that are brought from the periphery reach the central nervous system. Some of that information is stored as memory, and then a reply, a response is generated depending on where the signal ends up. So the ear, nose, eyes, etc. are considered external sensory function. They're receptors. So in the ear, the receptors in the ear are known as hair cells. And these cells are called hair cells because they have cilia and they swim in water. And if there's a bang in the tympanus, then the water inside makes waves and activates the hairs of the hair cells. In the eye, the receptors are the photoreceptors that are activated by photons, and so on and so forth. Can you explain, give examples of internal sensory receptors again? For example, receptors for oxygen, receptors for carbon dioxide, receptors for pH that can sense changes in the levels of these factors. Okay, so then we go to the integrative, that is the central nervous system made up of the brain and spinal cord. Now here, once the central nervous system interprets the signal, generates a signal that is now conducted by the motor part of the central nervous system to what we call the effectors. You remember these names: receptors, control center, and effectors. So integrative components are the CNS, yes. So here now the motor conducts the action potential to the effectors. So that means they do something, effectors. And those are the muscles, smooth, cardiac, and skeletal, and then the glands. So remember that the circuit is changes inside and outside. The receptors perceive. If these changes are strong enough, they generate an electrical current that is transmitted to the brain, to a specific area in the brain or spinal cord, and then a message is generated, another action potential is generated that is conducted to the muscles or glands via the motor component of the nervous system. Okay, so here, this is the motherboard of a computer. It's very simple, just to show you how this is so simple compared to the circuitry in the brain. So you have all these millions, billions of neurons all connecting with each other, making synapses with each other in a very regulated and organized manner. So it's very complex. Okay, so here's the organization of the central nervous system. Oh, sorry, of the nervous system. So this area here in blue corresponds to the central nervous system. Remember the integration. In green is represented the peripheral nervous system that has the sensory and the motor component. So we have here integrative, the central nervous system, sensory and motor, the peripheral nervous system. Which is the difference between nervous system and central nervous system? So the nervous system is the umbrella under which the central nervous system and the peripheral nervous system reside. So nervous system is the umbrella, then you have the central nervous system and peripheral nervous system. So here you see the central nervous system is made up of the brain and the spinal cord. And here you see the peripheral nervous system with the sensory neurons, with the receptors. So here is the sensory neuron. This is the cell body. The sensory neuron conducts the action potential towards the central nervous system. Sometimes there's an intermediate neuron we call the interneuron. This interneuron here in red is part of the central nervous system because it's located within the brain or the spinal cord. And then you see the motor neuron bringing the information out. Now, notice something very important here. Where are the cell bodies of these neurons? So this is a very, very, very simple circuit. You have the sensory neuron, an interneuron, and the motor neuron. So you see that the cell body of the sensory neuron is outside the CNS. You see, this is the cell body of the sensory neuron. Now, the cell body of the interneuron and motor neuron, these are located within the central nervous system. So they're located within the brain or the spinal cord. Okay, so now here you see a more complex vision of this. You see here in pink, the central nervous system, the brain, and the spinal cord that is lodged within the vertebral column. And now here in yellow, you see the sensory and motor neurons that serve or reach every part of the body. So the interneurons are these small neurons that are located within the central nervous system and that allow sometimes a sensory neuron to communicate with the motor neuron. Actually, most often there is always an interneuron, one or more, between the sensory and the motor. Okay, here's another view. The central nervous system in blue, and then in purple, the sensory and motor neurons that make up the peripheral nervous system. Okay, so now let's see what happens. How does this electrical signal, how is it generated? Okay, so here you see a candle with a flame. That's a stimulus, right? Heat. So where are the temperature receptors? They're located in the skin. The temperature receptors for heat, they are at the periphery. So they would be spread here, spread out through the hand. And now if you put a finger next to the flame, when it reaches a certain temperature, when that receptor is activated to a certain level, it will generate an electrical signal, an action potential. Here in yellow is the sensory neuron. So it's going to be conducting the action potential throughout the arm, and then it's going to go to the spinal cord. So follow the route here, and you see that the cell body is going to be outside the spinal cord. You can't see here very well. When it's the video, you'll see better. Then now the electrical signal is inside. This is a slice of the spinal cord, so a cross section, a slice of the spinal cord with the gray matter inside and white matter on the outside. So then the action potential is conducted to the motor neuron here in blue. See the cell body inside the CNS, inside the spinal cord, and the action potential is going to be conducted to the muscle, to the biceps. And it's going to allow the arm to move away very fast. So this is a reflex because it might not go to the brain because you need to have a very quick response. So it just travels to the spinal cord and right back out to the muscle. So this is just to show how the action potential can travel from the periphery through the sensory neuron to the central nervous system, in this case, the spinal cord, and then right out through the motor neuron here in blue to the muscle. Let's take a look. So you see the yellow? Here is the cell body of the sensory neuron, then goes through the interneuron, and then now the motor neuron is going to be activated. Now the biceps is going to be activated also, and will contract, and the hand is moved.
Professor, is the cell body of the sensory neuron on the hand?
No, the cell body is right here. In the hand is the receptor. The receptor is on the hand, and it's going, once that it's activated, once the sensory neuron is activated, here's the cell body. It's very close to the spinal cord. It's known as a dorsal root ganglion.
Oh, it's outside the spinal cord, right?
It's outside the spinal cord.
Okay,
Exactly.
thank you.
You are
Thank you.
absolutely correct. Okay. So the so the question is, so when there's some sort of stimulus, both the CNS and PNS are activated simultaneously. Well, milliseconds apart, right? Because first, what you see is activation of the PNS, the peripheral nervous system. And only when it gets to the CNS, then is the CNS. When it gets to the spinal cord, then the spinal cord is stimulated. Okay? So peripheral, milliseconds, central nervous system, and then back to the peripheral nervous system. So what is the ganglia? Here's a ganglion. Ganglion, singular, ganglia, plural. So ganglions are structures, stop. Ganglia are structures that contain the cell bodies of neurons located outside of the CNS, okay? So if the cell body of a neuron is inside the CNS, that's not called a ganglia, that's the CNS. Here, the cell body of this sensory neuron is located outside of the spinal cord. And here's is only one, but in this ganglion, there would be many, many, many cell bodies of the sensory neuron. And this area here that contains the cell bodies of the sensory neurons is a ganglion. Okay? So we'll move on. Okay. So now we go here. So what does this slide show you? This slide is comparing a human and a rat brain. Well, for the rat and the spinal cord. So the CNS of human compared with the rat. So here's the brain, human brain, and here's the spinal cord. You see all the neurons. The sensory and motor neurons are part of the spinal cord. And here you see the rat brain and the rat spinal cord. It's a big difference in size. Now, what happens here, what is interesting about the human brain, is that this area is known as the frontal cortex. And this is the site of intelligence. The frontal cortex is the area in the brain that is dedicated to intelligence. And you see what happens in the human brain is that the frontal cortex grew so much that it enveloped the other parts of the brain. And it grew so much that to fit in the cranium, it had to be folded. That's why the human brain, you see folds and crevices, because the frontal cortex grew so much. And there's explanations for this. One hypothesis is that when humans discovered fire, the fire could be used to break down food molecules and food particles. So cooking the food requires much less energy. Cooked food requires much less energy to digest than raw food. Because raw food, you have all the proteins still structured, so they have to go into the stomach and the acidity. breaks apart the bonds, and then they can be digested with the digestive enzymes in the stomach. Now, if you cook food, then the proteins are already going to be denatured, many of them. So when they get to the stomach, the enzymes can already chew on them. So, I mean, this is a very simple way of explaining this. So the fact that we cook our food, that means it requires much less energy to digest our food. And that excess energy was used to develop the brain. I mean, that's a pretty interesting kind of hypothesis or theory. So it's saying that we owe our intelligence to the discovery of fire. Well, you can accept it or not. What's the optics behind the sensory neuron cell body being outside? That's very good kind of question. Yes, so why is it outside? Because it can be regulated by many different factors, hormones, and so on and so forth. That's a very good question. So it has to do with regulation. Okay. So, next slide. So this slide is showing a real brain looking at the bottom of the brain. And you see here these white structures. These are cranial nerves. So here would be sensory neurons, and the motor neurons would come out of the brain through these nerves. So nerves sometimes can be mixed nerves. That means they have the sensory and the motor component. Sometimes they're only sensory. Sometimes they're only motor. So you see here, there's 12 cranial nerves. And here's the brain, and here's another view of the brain, more of a back to front. And you see the spinal cord, and you see the spinal nerves. When we talk about nerves, that means we're only talking about the sensory and motor, the axons, the sensory and motor components. So we have 12 pairs of cranial nerves and 31 pairs of spinal nerves. How was the nervous system extracted after death? It is not life, okay? This is after post-mortem. Okay. Okay, so now let's see here the components again of this circuit. So here you see in the peripheral nervous system, here you see a receptor. This is a piece of skin. Let's say it has the sensory receptors. Remember the Merkel, Pacinian, and so on receptors, and temperature receptors, and so on. So one interesting thing is that the receptors will not generate an action potential with any level of stimulation. It has to be a strong level of stimulation. Why? Because we don't want to be responding to any change. If we would respond to all changes from small to big, we would be a nervous heck. So we only respond, the action potentials are only generated when the stimulus is strong enough to activate, to generate, to trigger an action potential. And you will study this. So the receptors, once they generate an action potential, then you see that the action potential travels down the sensory neuron. Here is the cell body in the ganglion. And now it's been conducted to the spinal cord. So you see, here's the ventral root, and this is the dorsal root. So here, the sensory neurons go into the spinal cord through the dorsal root. That means it towards the back. Then the spinal cord you see here that it's always represented with a light color and a dark color. What does that represent? The dark color represents the area of the spinal cord that has cell bodies of neurons or interneurons. The white represents the area of the spinal cord that has processes, because some of them are myelinated. Remember myelin. So myelin is whitish. So that means that the outside is whitish because of the myelin that covers the sensory or motor neurons. The gray matter inside the spinal cord represents the area where cell bodies and some myelinating neurons, minor, but where the cell bodies are located. In the brain, it's the opposite. The gray matter is on the outside, and the white matter is in the inside. Okay, but now we're talking about the spinal cord. So now here you see the interneurons. So what is the beauty of having interneurons is that the signal that is coming through the sensory neuron is transmitted to this side, the same side. We call this the ipsilateral side. So if you want to move, the signal comes in and you want to move the leg on the same side through that corresponds to where the signal came in. So the same side, the ipsilateral. But the action potential can also be conducted to the other side, which we call the contralateral. So if we have here interneurons that would conduct the signal to the other side, that's through the interneurons. That's the beauty. One of the functions of the interneurons is to allow the signal to be spread out to different areas. Okay, so we talked about the receptor, the sensory neuron with the cell body and the ganglion, then synapses at the spinal cord with the interneuron synapses with the cell body of the motor neuron in the spinal cord. The motor neuron comes out and goes to the effector, which is the muscle. So this is the whole circuit. Receptor, sensory neuron, which would be the afferent pathway, if you remember, conducted to the central nervous system. And then we have the efferent pathway, the motor neuron, that brings the signal to the muscle. Okay. Now, this can be very complicated. Be prepared. How do we divide the peripheral nervous system? The central nervous system is very easy because we just have the brain and the spinal cord. That's it. Okay, now what happens to the peripheral nervous system? So in the peripheral nervous system, you have the sensory division, right? Which is this part that brings the signal to the central nervous system. And there is the motor division, which brings the signal outside. Okay. So the sensory division can receive signals from organs, right, inside our body. Those are called viscera. Viscera means the organs inside. So the sensory division would bring signals from receptors located in the viscera to the CNS. If there are receptors like these that correspond to the senses and the cutaneous receptors, they go through the somatic sensory division. So these are autonomic, we have no control. And these are going to lead to the skeletal muscles, to the muscles, skeletal muscles. So there's two divisions within the sensory part of the peripheral nervous system. So we're going from peripheral to sensory to visceral and somatic. So this is going from the outside to the inside. Now let's look at the other side, going from the central nervous system to the periphery. That's the motor division, going from the central nervous system to the periphery. So there's also visceral and somatic. Visceral are the signals that are conducted to the organs. So there's two divisions here, sympathetic and parasympathetic. Sympathetic, alarm, fight or fight, and parasympathetic, slow, homeostatic. And then we have the somatic that will conduct the signal to the skeletal muscle. So from the senses, you go to the central nervous system and to the skeletal muscle. So you go somatic sensory, CNS, somatic motor. From the organs, you go visceral sensory, CNS, and visceral motor, and then sympathetic and parasympathetic. So here the effectors of the visceral motor division are smooth muscle, cardiac muscle, and glands. The effectors of the somatic motor division are the skeletal muscle. So complicated, but if you think about it, it's logic. Now, always remember anything that's somatic, it's under voluntary control. Our mind controls it. Anything that is visceral is autonomic. That means we don't voluntarily control. And then there's the enteric nervous system that is separate from all this, that has 500 million neurons dedicated to the digestive tract. Okay, so this is a little bit complicated. Okay, now let's go into more detail. Let's look at the sensory and motor neurons. So this was sort of the geography, the organization of the nervous system, that umbrella, the nervous system, divided into central and peripheral. Central, very easy to remember. Peripheral, a little bit more complex. Okay, now let's go to the neurons. What do they have in particular? Up here, this is the spinal cord. Remember the gray matter in the center. So this is a sensory neuron. Here would be the receptors. So the sense organs, these would be the receptors. The action potential is traveling. Here's the cell body in the ganglia outside the spinal cord, and then continue on and synapsing within the CNS. Here you have now two different types of motor neurons. These two lavender neurons are motor neurons. You see the cell bodies inside the spinal cord, right? One motor neuron, another motor neuron. Now this motor neuron is part of the somatic division. So going somatic, we're talking about here. So the somatic motor neuron brings the action potential to the skeletal muscle. And now you see that this is continuous, right? This is one motor neuron going from the spinal cord directly to the skeletal muscle. Now remember the sciatic nerve, I don't know if you ever heard of it. It's very, very long. It's the longest nerve in our body. It goes from the spinal cord all the way down to the tippy toes. One axon, one whole axon. And that's why sometimes it's impinged and it causes a lot of pain. So one motor neuron for the somatic division. What about now the autonomic division? Now we're talking about visceral. How is the visceral motor neuron? Now you have two motor neurons. One has a cell body in the spinal cord. The other one has a cell body outside. So this would be a ganglion, right? Because there's a motor neuron outside the spinal cord. But here, we're not talking about sensory, we're talking about motor. So the preganglionic motor neuron has the cell body in the spinal cord. The postganglionic neuron has the cell body in the ganglion. So you see here now that for the viscera, there's two motor neurons in tandem that are conducting the action potential from the spinal cord to the periphery to smooth muscle, cardiac muscle, and glands. And all this, and you might think, wow, why is this organized like that? Why do we have only one motor neuron in case of somatic or skeletal muscle voluntary, but we have two for the autonomic? So this is a way to not have so many cell bodies of motor neurons within the spinal cord. That would be chaos. So like this, we're pushing some of the cell bodies outside of the central nervous system. Okay, so here we have sensory going from the periphery to the center with the cell body outside in the ganglia. Then we have the somatic motor, just one motor neuron with the cell body in the spinal cord. And for the visceral motor, we have two motor neurons. The preganglionic with the cell body in the spinal cord, the postganglionic in the ganglion outside the spinal cord. Okay, so this is another view. You see here the spinal cord, and here we're showing the two motor neurons, or the structure of the two motor divisions. So here in the autonomic, we have a preganglionic and a postganglionic. The preganglionic brings the action potential from the spinal cord. See the cell body in the gray matter of the spinal cord, and its synapse is at the ganglion, an autonomic ganglion, where the postganglionic neuron is located, starting the cell body, carrying the action potential to smooth cardiac muscle and glands. Now here's the somatic nervous system, the motor component, only one motor neuron, going from the spinal cord, the cell body in the spinal cord, the gray matter, going directly to the skeletal muscle. So very quick action potential being conducted here. Okay, now let's add another layer of complexity. So we're still talking about somatic versus visceral. Okay, remember, here's somatic 1, visceral 2. Somatic motor neuron, it's myelinated. So you want to be able to control your skeletal muscles very quickly. So you only have one motor neuron. Besides that, it's myelinated. So that means the action potential goes very quickly. 70 to 120 meters per second, like a race car, faster than a race car. Very quickly. Just remember putting your hand on that flame. You want to be able to take it away very quickly. If not, you will get burned. So that's why you have one motor neuron. And it's myelinated. And what is the neurotransmitter that is being released here? You already know the answer. Acetylcholine. So that means that this neuron, we call it cholinergic neuron. Why? Because the neurotransmitter that is stored at the axon terminal is acetylcholine. Now let's see down here where we have the two motor neurons. So in the visceral or autonomic nervous system, here's the first motor neuron is myelinated. So the preganglionic neuron is myelinated and it's also cholinergic. It releases acetylcholine. But now, note this, the postganglionic neuron is unmyelinated. So you want the response to be more slower and more and stay for a longer time. So it's unmyelinated. And now what are the neurotransmitters? You can have two types of neurotransmitter, either acetylcholine or norepinephrine. This is another neurotransmitter that you haven't heard yet. So the motor, the The somatic motor neuron releases acetylcholine, and so does the preganglionic motor neuron of the autonomic nervous system, acetylcholine. Now, the postganglionic can release acetylcholine and or norepinephrine. So why is this all important? Because now you can have drugs, for example, because we know norepinephrine is released in some of these post-ganglionic neurons, we can design drugs that affect just the preganglionic neuron and not the others, because they will bind to norepinephrine receptors and not acetylcholine receptors. So NE, yes, thank you, stands for norepinephrine. What happens if the autonomic had the myelination in the pre and post? Well, this response doesn't need to be so fast because you're affecting, glands and smooth muscle, cardiac muscle. So it's slower. The response should be slower. So you're putting a brake by not having it myelinated. Okay, look at the difference in the speed. 70 to 120 meters, 0.5 to 2 meters per second. Okay, so here is more complicated now. Let me see how many slides I have. Oh, we're almost there. So now let's compare the organization of the parasympathetic and sympathetic. So this part here. So remember, there is sympathetic and parasympathetic. One is activated under alarm conditions, the other ones when we are relaxed. Okay, so on this side is the parasympathetic and here's the sympathetic. Now what we're looking at is the organization of the pre- and postsynaptic neurons. Some of them are longer, some of them are shorter. So let's see. In the parasympathetic, you see that the preganglionic is very long and its synapses very close to the organs. You see the presynaptic in red, the postsynaptic in green. So the synapse, this ganglion, is very close to the organs that are going to be innervated or stimulated. On the other side, the sympathetic, you see the same organs. So the same organs can receive parasympathetic or sympathetic stimulation. And I will give you an example. So here you see that in the sympathetic division, the preganglionic is very small, very short. And the ganglia are arranged in this chain, sympathetic chain, that is parallel, it's outside the spinal cord, but is parallel to the spinal cord. So you have very short pre-ganglionic and very long post-ganglionic. Would you mind going over what a ganglia is again? A ganglion, ganglia is plural, ganglion is singular. A ganglion is a structure that contains cell bodies of neurons that are outside the CNS, outside the central nervous system. So you see this ganglion contains the cell body of the postganglionic fiber. This is outside of the spinal cord. That's what a ganglion is. So here you see a chain of ganglia, the sympathetic nervous system, but here the ganglia of the parasympathetic are located near the viscera. So its synapse is inside the ganglion. Exactly. Perfect. Yes. Okay. Now, just one more complexity. Oh, you already know this. Because what are the neurotransmitters? So here we're talking about the sympathetic. When you are in alarm state. So preganglionic acetylcholine, cholinergic neuron, at the ganglion, it releases acetylcholine. So that means that this cell body has acetylcholine receptors. But there are two types of acetylcholine receptors. Here, these receptors are known as nicotinic receptors. And when we go through these lectures, you'll understand the difference between nicotinic and muscarinic. So the nicotinic receptor responds to nicotine in cigarettes. So these receptors are nicotinic receptors, the receptors located on the cell body of the postganglionic neuron. Now here, this is the postganglionic neuron, and it can release either acetylcholine or norepinephrine. So some cells will have norepinephrine receptors or noradrenergic receptors. Other cells will have cholinergic receptors, but in this case, they're muscarinic. So the target cells either have muscarinic or adrenergic receptors. Muscarinic respond to acetylcholine, adrenergic respond to norepinephrine. The cell bodies of the postganglionic neurons have nicotinic receptors. So nicotinic receptors here, the target cells, which would be smooth muscle, cardiac muscle, and glands, they either have muscarinic receptors or adrenergic receptors. And to know this is unbelievably important to treat patients, okay? Because you want to know what kind of receptors are on the cardiomyocytes or on the pupils or here and there on all these different organs. They all have different receptors, and it's very important that we know what they are so we can design drugs that are specific and will affect the heart and will affect the heart and will have minor effects on the lungs, or will affect the kidneys and will have minor side effects on the liver. So knowing these receptors for you might be a pain because you have to learn this language. After all, it's a language in human physiology. So, but it's very, very important because it's the way that we can design more and more specific drugs that will affect one receptor and not other receptors. So specificity is what is important here. So now the parasympathetic, you have acetylcholine at the released at the preganglionic neuron. The receptors are nicotinic. But now the postganglionic also releases acetylcholine, and the target cells have muscarinic. So the target cells, sympathetic or parasympathetic, they have the muscarinic cholinergic receptors, and the sympathetic have adrenergic also. The parasympathetic do not have adrenergic. Okay, so let me put this into perspective. This is my last slide. And then we can go over the exam. So here you have the central nervous system, the brain, and the spinal cord. Here you have a ganglia outside the spinal cord. And here's another ganglia. It's called the ciliary ganglia, and here's the superior cervical ganglia. So this yellow side here represents the sympathetic nervous system. So when you are scared, your pupil opens up. So how does that happen? Going from the brain, the signal, the alarm reaction, how does it end up with a widening of the pupil? But when we are relaxed, the pupil the diameter is smaller. So it's being activated by the parasympathetic nervous system. So you see another ganglion here. The blue is parasympathetic. Okay, now let's see what happens. So the action potential, now we're only considering the motor division, the action potential being transmitted from the central nervous system to, in this case, the eye. Okay, So let's see, you are all of a sudden you go to a room and there's a lot of light, and this activates the sympathetic nervous system. And now here you have the motor preganglionic neuron releasing acetylcholine at the ganglion. And now you have the postganglionic neuron that will release norepinephrine. So a different neurotransmitter. So that means that the muscles here, the smooth muscles, the radial muscles, will respond to norepinephrine when they are stimulated by the sympathetic nervous system. So that's how the pupil of the eye knows how to expand or to become smaller. It depends on what receptors and what neurotransmitters are released at the smooth muscle layer. So here you have the sympathetic nervous system releasing norepinephrine. The receptors are adrenergic receptors. When activated, they will contract and cause the pupil to dilate. So sympathetic nervous system, the result when it's activated is pupil dilation. And you have cholinergic neurotransmitter, but here you have norepinephrine. What about in the parasympathetic? So you have acetylcholine at the ganglion, and you have acetylcholine here at the pupil. Now, which smooth muscles are going to respond? The circular muscles, because they have the receptors for acetylcholine. So muscle constriction. So the radial muscles respond to norepinephrine. The circular muscles respond to acetylcholine. And that's how in one organ, one eye, you can get both responses when activated by the sympathetic or the parasympathetic nervous system. So most organs in our body receive message from sympathetic and parasympathetic nervous system. And the response that is generated depends on the neurotransmitter that is released at the postsynaptic neuron. This was complex, but I hope that you understand the importance of knowing all this. And we are going through this with this next set of lectures. So any questions about the material? I don't want to continue here. So, yes?
Professor, so for the sympathetic system, the postganglionic neuron releases norepinephrine and acetylcholine, both of them, right?
No. You almost said it correctly. I was so happy. Here, oh, you mean in general, not on the eye. Is that what you were saying? Then you were correct.
Yeah, for general sympathetic.
Yes, general sympathetic can be either norepinephrine or acetylcholine. Yes, absolutely.
From the postganglionic neuron, right?
Yes.
And for the parasympathetic, it only releases acetylcholine.
Perfect.
All right, thank you.
Yes. Okay. Yes. To be clear, the lecture notes today is only one page long. Yes. But look at all the material that I covered, right? So you have to really understand this. And you're going to have to learn this as we go on. Okay? So yes, it's only one page. You're absolutely correct. Okay, so can we move on to the exam, or you don't want to look at the exam?
I will cry again.
So you don't want to look? Nobody? No voluntaries? Nobody wants to look at the exam? You want to look. Okay. Let's see if I can-
Professor, one more thing. The last exam is not going to be cumulative, right? It's only nervous system?
Yes, absolutely.
Okay.
Oh no. Give me a minute. Hmm. Exams. Two. Okay. So now we go here. Share. Okay, can you see, or let's see if I can make it bigger.
Yeah, I have to zoom in more. Let's see.
I have to zoom. Uh, um, let me see. Let me stop share and let me do this. Oh, that's good. I did it. Okay. Now let's go here. Okay, now you can see, correct? Yes? Hello?
Yes.
Oh, I see. Okay, so here, let's go to the first one. A 75, so you guys ask me questions, or do you want me to read and then we together find out what is the correct answer? A 75-year-old woman becomes bedridden after hip surgery. After 6 weeks, imaging shows decreased bone density in the femur. Which mechanism best explains this finding? Someone tell me.
A.
Yes. Reduced osteoblast activity, right? Triggered by the lack of mechanical stress. Perfect. Let's go to the next one. Is this a good style, a good idea to go like this? So there's a question here. The length of pro and post ganglionic ganglia of the pre and post ganglionic neurons, does it have anything to say about the speed the effectors can receive the information? Yes, and specifically the myelination, right? Okay. So 2, excessive, a pediatric patient has a fracture through the epiphyseal plate. The epiphyseal plate allows growth. Which consequence is most likely? Excessive width. No, if it's fracture, it's non-functional. So what would be the correct answer? B, correct. Okay, next one. Oh. Which sequence correctly describes endochondral ossification? Okay, so endochondral ossification starts with the hyaline model, right? The cartilage model. So that's the first thing. So you look down here, there's only one. Which correctly describes endochondral ossification. So which one?
B.
B, right? Because you have mesenchyma. That's undifferentiated, right? So mesenchyma, cartilage, primary ossification, and cavities. Perfect. 4, blank is a congenital defect caused by failure of the vertebral lamina to unite at the middle. It can be associated with, so, spina bifida and folic acid, right? None of the others. It's a congenital defect caused by vertebral lamina not uniting, right? So the answer is E. Okay. On a field trip, you discover a skeleton with the following characteristics. Overgrowth of only the hands, feet, and lower part of the face. This skeleton is probably from an individual suffering from acromegaly. This is when growth hormone happens after puberty, if you recall. Okay, next one. A patient undergoes total thyroidectomy, so thyroid removed. Parathyroid glands are preserved and function normally. So you know that parathyroid glands are essential for calcium. Thyroid is not so much. Calcitonin is not so important. Which change in calcium homeostasis is most likely immediate? Goes thyroidectomy for cancer. Parathyroid glands are preserved and function normally. Which changes in calcium homeostasis is most likely? Which? Oh. So, life-threatening hypercalcemia, no. Little to no change in calcium levels. That's the correct, right? B, because you still have parathyroid, so who cares about the rest? Okay, 7. A 7-year-old with dietary vitamin D deficiency presents with bowing of the legs. Which process is least affected? Well, deposition of calcium is it going to be affected? Maintenance of serum calcium through intestinal absorption, yes. Bone flexibility, yes. So which process is least affected, right? All these so far are going to be affected. Integrity of mineralization bone matrix, yes. So E, the increased osteoblast number. Vitamin D has nothing to do. Samia, here you are. The E, increased osteoblast number, has nothing to do with vitamin D. All these other factors do. 8. Which sequence correctly reflects the lineage of both bone-forming cells in development? Okay, you know that the first thing is mesenchyma, osteogenic cells, osteoblasts, osteocytes. So you can immediately eliminate others and know that C is the correct one. 9. Excess growth hormone prior to puberty would result in gigantism. This is easy. A patient has high parathyroid hormone. Which bone cell activity will increase first? Remember, parathyroid hormone is activated, or the parathyroid is activated when there's low calcium. So the parathyroid hormone, its effect is to increase calcium. So what would be activated? The osteoclast, because the osteoclast releases calcium from the bone. C would be the correct. John is suffering from leukemia and is going to receive a transplant from Tony, his healthy cousin. Which of the following would provide healthy blood cells? Okay, the precursors are in red bone marrow. So B is the correct answer. A long distance swimmer, so non-weight bearing exercise, right? Because it's just the weight of the body, has lower bone mass than a cyclist who also does weightlifting. Which principle explains this difference? Can you explain why other answers are? You want me to explain why other answers are incorrect? For all of them or for any specific question? Which question are you talking about? Because if I'm going to do this, it will take me forever. For the last one. You mean number 11, yes? 10. No, 11. 10. A patient has high parathyroid hormone. Which bone cell activity will increase first? It has to be osteoclast to release calcium. Osteoblast puts calcium on the bone, so it would even go worse. It would be the parathyroid does not activate osteoblast, does not activate osteogenic stem cells, does not activate osteocytes, and does not activate chondrocytes. It activates osteoclasts. Those are the ones that have the receptors. Okay, number 11, the red bone marrow. Okay, now we were here. Long distance swimmer has lower bone mass than a cyclist who also does weightlifting. So weightlifting will activate osteoblasts. So which explains this difference? Bone remodels B, right? Bone, the Wolff's law, bone remodels primarily to weight-bearing stress. That's the correct answer. Okay. 13. Do you want to continue? When you want to stop, let me know. I'm fine. I can stay. But if you want, let's see, how many people do we have? 34. Okay, it's fine. 2y for staying. Oh. Thank you. Okay, I have to understand what TY means. Okay. During experimental inhibition of osteoclast function, serum calcium drops. The inhibiting osteoclasts. So the osteoclast cannot do resorption. So why? Osteoclasts secrete, osteoclasts activate, osteoclasts normally release calcium. A is the correct answer. Oh, okay. 14. A patient develops patches of bone formation inside skeletal muscle. You know that this is not intermembranous ossification, calcification, conversion of yellow marrow, intermembranous ossification in connective tissue. So that means it's forming outside the skeleton. So it's forming in muscle. So that's intermembranous ossification in connective tissue. That's an easy answer. A tibial shaft fracture is casted. 2 weeks later, X-rays shows a bridging, low density mass connecting fragments. So the fragments are connected by low density mass. So that means it's not bone yet. So if it's not bone, it's not hematoma, because hematoma would not connect. Dense compact bone, fibrocartilage, soft callus. That would be the correct answer. C. Right. C. Fibrous cartilaginous soft tissue. Because this doesn't exist. Lamellar bone remodel, medullary cavity fully reestablished. The only one that makes sense is number C. Soft callus. Okay, number 16. A non-union fracture is treated with pulsating electromagnetic fields. The most plausible cellular explanation for benefit is not activation of osteoclasts. Attraction activation of osteoblasts by stress generated electrical fields. That's correct. Oops, B. Okay. 17. Oops. A patient with long-standing hyperparathyroidism hyperparathyroidism. Remember, hyperparathyroidism increases osteoclasts, so has bone pain and deformities. What skeletal change most directly reflects excess parathyroid? So if you're releasing too much calcium, the bone is going to be less dense. It's going to be demineralized. So B would be the correct. A patient with renal failure has low calcitriol. Calcitriol is the active form of vitamin D. Predict the compensatory endocrine response and its skeletal consequence. So, if a patient has renal failure, has low calcitriol production, so now probably has low calcium, right? If it has low vitamin D. So you want to, the compensatory mechanism has to be to increase calcium. So, which one would be the correct one? Decrease PTH, so it has low calcium in the blood. So you want to increase calcium levels. So B would be the correct. You want to increase osteoclast activity to increase calcium in the blood. Right, Samia. So you guys get it? So if you have low calcitriol, that means you would result in low levels of calcium in the blood. So the compensatory mechanism would be to increase blood calcium levels. You would have to have high PTH to increase osteoclast activity. A 62-year-old postmenopausal woman with osteoporosis refuses estrogen therapy because of concern with breast cancer, but wants a medication that provides vertebral fracture protection. Which therapy is most appropriate? Well, you can give her estrogen. So would you give her raloxifene? Why is 18 not C? Because calcitonin increased osteoblast activity. Calcitonin increases osteoclast activity. Sorry, not osteoblast. Because calcitonin, you want to increase calcium. Right. So, this postmenopausal woman, you wouldn't give her estrogen. You would give raloxifene for vertebral fracture protection and anti-estrogen effects. This is the most safe for increased bone density. Okay. Number 20. A healthy 54-year-old, 3 years post-menopause, shows early osteopenia on DEXA. Which primary mechanism underlies her declining bone mass? B, estrogen decline, correct. Estrogen, so it's 3 years postmenopausal, has less estrogen, and therefore the osteogenic cells have receptors for estrogen. So you have more osteogenic cells, you have more osteoblasts, you have more calcium being deposited on the bone. So the answer here is has estrogen decline. When a surgeon repairs a torn ACL, the new B is correct. 20B is correct. The new ACL is created using tissue from a donor, such as an Achilles tendon. The tissue source, so it's from a donor, it's not their own. So the tissue source is called an artery, and the procedure is arterioscopically. So which one is it? It has to be the allograft, right? B. Carpenter has Lateral knee pain when squatting. MRI shows fluid in a sac outside the joint. So that has to be a bursa. If you have fluid in a sac outside the joint, is a bursa. So C would be correct. Why does moderate repeated joint movement protect joint health in freely movable joints? So what it does, it forces, no, the blood vessel, it pumps nutrients, rich fluid through the joint space. Remember that cartilage has no blood, so it's relying on the synovial fluid, and movement allows the synovial fluid to hit the cartilage and feed it. How many people do we have now? 23. Okay. A 71-year-old reports grinding pain. So that we can already know that it's osteoarthritis because it does not have inflammation. During vigorous running, synovial fluid briefly becomes highly viscous. So you know that that is to protect the joint by acting as a dilatant shock absorbent. I emphasize this about the dilatant property of the synovial fluid. "26. A slip disc most often refers to complete tearing of annulus fibrosus with extrusion of the nucleus pulposus. Yes, A is correct. You following me? Nobody wants to tell me the answer? Okay. A patient with severe rheumatoid arthritis develops pannus. Remember, this is inflammation caused by it's an autoimmune disease. So what do you want? How can you treat the autoimmune disease? With immunosuppressants, right? That's what we use. C. 28. The procedure for examining and repairing the interior of a joint with minimal incision. Well, this question was one of the questions I gave you. So it's arthroscopy. A 54-year-old obese patient develops gouty arthritis. So what do you mean? Uric acid crystal deposition. Gouty arthritis is caused by uric acid crystals. Rising onto the toes, what kind of lever is that? Second, right? Good. Okay. During excitation of a skeletal muscle, depolarization is initiated when, well, immediately you know that it's voltage-gated sodium. So you could go right away to A. Right? So the depolarization is caused by voltage-gated sodium ions. So A. What was 30 again? So second class level. So here, you know that it's sodium, so you could go right away to A. A genetic defect prevents the dihydropyridine receptor from functioning. Which downstream event fails first? Calcium liberation to cytoplasm. So going from the sarcoplasm to the cytoplasm.
Professor, I had a question about the
Yes.
voltage gated one. I thought in lecture you had said that it's initially like a sodium ligand gated channel, and then the change in voltage triggers the rest of them.
That's
that are
right.
voltage.
That's why I say A.
Isn't the first one like the very first channel that opens, isn't it ligand gated?
Oh, no, but that's the depolarization, right? Because remember, when it's at the motor end plate, you only go from -90 to -60. When you start real depolarization, is when the voltage gated. You know, when you go up the curve, the spike, that's the depolarization. That's when the sodium, when you go from minus 60 to plus 30, that is the depolarization. So the trick here is depolarization. Okay. But anyway, where would you see here ligand-gated sodium? There's no option for ligand-gated sodium.
I know, that's why this one kind of tripped me up a little bit because I thought
Okay.
that was the initial one.
Yeah, but if it would be that, it should be sodium, right? Not potassium.
Right.
Okay, so for 32, the genetic defect prevents dihydropyridine from functioning. The answer is C, calcium liberation to cytoplasm. Now, 33, a teenager presents with progressive muscle weakness. Biopsy shows absence of linkage between actin and the sarcolemma. You know that that's muscle dystrophy. It's dystrophin. The protein that links actin, the fin filament, to the sarcolemma. The release, DHP is dihydropyridine. Dihydropyridine is the receptor that communicates, same as nicotinic, no. Dihydropyridine is a receptor that communicates with the calcium channel on the sarcoplasmic reticulum. Okay. The release of calcium from the blank exposes myosin binding sites on actin by way of the, so, release from where? The release of calcium from the, so what would be? The terminal cisterna, the sarcolemma, myosin I bands. Right. Remember what the terminal cisterna is. That's the sarcoplasmic reticulum that is on each side of the T-tubule. Okay. A toxin prevents synaptic vesicles from fusing with the motor terminal membrane. Which downstream effect fails first? So you have no synaptic vesicles fusing, so there's no ACH being released. So what would be the effect what would be the first thing to happen? This one, binding of acetylcholine to the receptors because there's no acetylcholine. A fine motor movement, like controlling the vocal cords, is lost after a microstroke damaging one motor neuron. Which fact explains the severity of precision loss? So motor units in larynx contain only a few fibers per neuron. It's very precise. A whole muscle is stimulated at increasingly higher frequency until individual twitches fuse into a smooth plateau. So what would that be? Tetany. Which sequence correctly describes the events at the neuromuscular junction immediately before an action potential is initiated? Now here you have, so what happens? Which sequence correctly describes the events at the neuromuscular junction immediately before an action potential is initiated? So ACH binds its receptor. Okay, calcium enters the axon, synaptic vesicles. So the first thing is calcium enters the axon terminal. Then the synaptic vesicles fuse and exocytose acetylcholine. Acetylcholine binds its receptor and then opens in sodium influx. So we'd have 2, 3, 1, 4. 2, 3, 1, 4, 8. I thought none of them was right. Oh, okay. So the calcium enters the axon terminal. Remember, that's the first event, the signal for the synaptic vesicles to move and fuse with the plasma membrane at the axon terminal. So after knowing that, it could only be A or E. So then the next step is that the synaptic vesicles fuse. So that would be 2, 3. So then you would go there. I think the confusion aspect was that calcium is used in 2 different places. Yes, that's true. One for the axon, at the axon, and I emphasized that in the lecture actually, that calcium is used in 2 sites. 39. Place the following in correct order. ACH is released into synaptic cleft. Muscle action potential travels along sarcolemma. Dihydropyridine receptors activate and sarcoplasmic reticulum open and release calcium. So what happens first? ACH is released. Muscle action potential travels along sarcolemma. ACH is released into synaptic cleft. Then the muscle action potential travels along the sarcolemma. Then, so it's 1, 2, 3, 4. A is the correct one. So the first is that the acetylcholine is released into the synaptic cleft. During contraction, sarcomeres shorten because slide towards while the actual length of the thick and thin filaments does not change. Sarcomeres shorten because which one should be? Should it be C or D? So what slides is the thick filament? Or is it the thin filament? Sarcomere shortened because slides toward the length of the thick and thin filaments. Do not change. So does not change. It can only be C or D. So the thin filament slides towards the M line, right? The M line is what keeps the thick filament in place. So the thin filament slides towards the M line. So C would be the correct. You guys agree? It's a hard question, but it's doable. Rigor mortis, I have to have 1 or 2 or 3 questions that are a little bit harder. Rigor mortis causes sustained contraction, primarily because after death, the cross bridges don't come apart, right? IATP is absent and cross bridges cannot detach. 42. Severe hypocalcemia can produce tetany because low calcium in plasma prevents calcium from binding, blocks ATP, increases sodium channel opening and cell excitability. So tetany is cell excitability. So C is the correct answer. Do we still have people or I'm the only one left? Let's see. I think we still have a few. 17. Okay. Relaxation of skeletal muscle begins when ACH is degraded by acetylcholinesterase. So right away you can see B. Correct order with a single cross bridge cycle. So ATP binds myosin. Myosin head hydrolyzes ATP. Myosin binds to exposed actin sites and power stroke occurs. So 4 has to be the last one. Let's see, ATP. I'm getting a bit tired now. Correct order within a single cross bridge cycle. So 1 seems to be the last one because ATP binds and cross bridge detaches. So 1 is the last one. So it can only be B or C. So myosin head hydrolyzes ATP. Myosin binds to exposed. So it has to be myosin head hydrolyzes ATP. So from these two, let's see if this is correct. E. Myosin head hydrolyzes ATP, yes. And then ATP binds myosin and cross. No. Where were we? This one is not. Let's see. Myosin head hydrolyzes with a single cross bridge. Power stroke occurs, PI released. So ATP, so which one? Help me out, guys. ATP binds myosin and cross bridge detaches. No. Which is the first step? Myosin head hydrolyzes ATP. 2. Myosin binds to expose actin sites. Yes. Yeah, this is it. 2, 3. Myosin head hydrolyzes ATP. Myosin binds to expose actin. Power stroke occurs. And then it's 2, 3, 4, 1. It's B. I guess you are as tired as me. You want to continue to the end? Yes, let's go. A sprinter explodes off the standing blocks for a 100-meter dash. So this, it's B, creatine phosphate. During moderate endurance exercise, oxygen continues to enter muscle fibers from hemoglobin. It's also released from myoglobin. That's easy. 48, B. 49. A highly conditioned marathon has a Vmax of 80, which is the most direct physiological consequence of this adaptation. B. Increased capacity for aerobic ATP production. 50. Heavy breathing persists after strenuous exercise because oxygen is needed to close voltage-gated sodium channels to destroy ATP in mitochondria. Yes, to convert lactate to glucose and restore creatine phosphate. So C is the correct one. A distance runner calf biopsy shows many mitochondria and slow fatigue. So that would be B, slow oxidative, right? Distance runners, they have many slow oxidative fibers because it's long run. They have to rely on aerobic ATP production. A physician explains to a patient that cardiac muscle cannot enter tetany like skeletal muscle. Why? Because the cardiac refractory period. Cardiac refractory period is long. Remember, the refractory period in the cardiac muscle is long to allow the ventricles and atria to fill in with blood. A clinician explains to a patient that the heart keeps beating even when all somatic nerves. Which cell property is the pacemaker? C. A patient with severe constipation is given a promotility drug to increase contractions that push food along the gut. Which smooth muscle property enables this way? Multi-unit innervation? No, single, right? Single unit smooth muscle innervation with many gut junctions. That's the the smooth muscle, how it's innervated in the gut. A physician explains to a patient, I don't know, what did I say? Is given a pro-motility drug to increase contractions that push fluid along the gut. Which smooth muscle property enables this coordinated way? The multi-unit is not, because it's coordinated. The key word here is coordinated. So that has to be the single unit smooth muscle. So where are we? Physician, okay, and clinicians. Oh, are we going to 55? A woman treated with Botox for facial wrinkles develops localized paralysis. Botox blocks smooth and skeletal muscle contraction primarily by blocking ACH release from motor neurons. Remember, it prevents the synaptic vesicles from fusing with the plasma membrane. Fill in the blank in smooth muscle. Calcium binds to calmodulin, which activates myosin light chain. B. In athletes performing repeated isometric lifts, so the size is the same. A bulge in the groin develops when viscera pushes through. So you know it is the inguinal canal.
Professor, I have a question about the Botox one. There was a
Yes.
video you posted in these slides, I believe, and it showed like a figure of the ACH receptors on the sarcolemma being blocked from the Botox. So how is it B for 55?
No. The ACH receptors, that's myasthenia gravis. The Botox, it prevents the synaptic vesicles from being released from the synaptic vesicles from fusing with the plasma membrane of the axon terminal. The receptors, it's an autoimmune disease. Okay? So you can go back and I explained well that what the Botox does, it prevents the acetylcholine from being released. It has nothing to do with the receptors. It's just the acetylcholine is not being released.
Okay, thank you.
Okay, sorry about that. Okay, calmodulin, we were there now. Okay, we were here, 57. In athletes performing repeated isometric lifts, a bulge in the, we did that one. The inguinal, inguinal now due to elevated pressure in the intra-abdominal pressure. 58. A patient's bladder fills slowly without large increase in pressure until threshold triggers voiding. Which smooth muscle property enables this? B. Stress, relaxation, and response. Remember, to allow the bladder and the stomach to fill, the muscles can be stretched without contracting right away. 59. Smooth muscle can regenerate better than skeletal muscle because parasites, right? And 60, sequence of events in tetanus infection leading, so tetanus, remember, it's caused by a bacterial infection, leading to sustained contraction. So wound entry of the bacteria. Then the toxin spreads to the central nervous system. Then it inhibits the GABA glycine release. These are the inhibitory neurons. And then you have sustained motor neuron activity. So you prevent the break. So it's 1, 2, 3, 4, 8. Oh, finished. Okay, guys, that's it. Stop sharing. We arrived to the end. And meanwhile, 6 o'clock. Okay. Any
Hi.
comments? Yes.
Yeah, Professor. I'm just talking about like me, like with the issue with making up the exam that I like emailed you about. I just wanted to
Yes.
ask, just really quickly that, um, I take it the final December 16th, so I'm taking two exams the
Yes.
same
Back to back.
in one day. Okay, that's a lot.
Back to back.
Okay.
So one is 1:45 to 2:45. The other one is from 2:45 to 3:45. And didn't I send you the grading policy? Anyway,
Yeah,
that's what it
you
is.
sent the grading policy. It's
And
just
it's
yeah.
also listed in the syllabus.
Yeah, I just wanted to make sure because
Yes,
it's
that's
a
what it
lot
is.
for me mentally. Like
Yeah.
we don't even get like a 5 minute break or anything. Like
Yeah,
just
you might get a 5-minute break.
okay.
Yes. Okay.
All right, thank you.
Any other comments? Believe me, there's many students like you because of the storm.
Professor, I have a question. I
Yes.
posted in the chat, but do you have like any recommendations or resources on how we can study for the exam? Because I'll study the lectures, the recordings, and the notes, and I'll feel like I have the science down pat. But then when I look at the exam questions, I feel thoroughly lost. So, is there anything you could recommend to understanding applications of the science we learned in class?
So there's lots of questions out there, and there's questions on mastering. And do you study the review questions also?
Yes,
Like,
I re-reviewed it right before the exam and I was like so confident. And then when I actually saw the questions, I was still like, I don't know, just like lost.
yeah, you have to know the material very well, right? But they're not hard questions because, you know, if you know exactly what you're looking for, you can pick the answer. And you don't, either you know it or you don't. They're not hard questions. I'm not trying here to trick anyone. And so there's lots of questions out there. Look for questions on the nervous system, right? That's the next the next exam. Look in mastering. Also look in the back of the chapters. There's a lot of questions there also that you can use. So the trick is to answer questions and to time yourself. Use a
Okay.
timer when. Go
Sorry.
ahead.
I know you provide like some review questions prior to the exam, but I wanted to ask because I went to City College before Hunter, and they also had like exams from previous years, like online that you can search up. Does Hunter do that, or do you have anything from previous
No,
years?
no, I don't have that.
Okay.
But there's many questions out there that I've been teaching this for a while. There used to be people that would take pictures of my exams and then sell them. So they might be out there. I don't know. Who knows what happens out there under? I don't have any control.
Um, I'm sorry, I have just one more question. So, if I bring like medical documentation, would I be able to like take my makeup exam to the day of the final, but take the final on the next day with the final makeup day if I have medical
No,
documentation?
no,
Because
no, because you already missed one exam. You can only miss one exam. That's it.
yeah, but it's
Look,
like
if
if I
you
have a
are
medical
healthy to take
reason.
one exam, why wouldn't you be healthy to take the other exam? It makes no sense. Okay.
No, it's just because like just two exams back to back, it's just a little
Well,
too much
many
for me.
students,
No, it's because like I would bring medical documentation, so I'm not like other students, if you know what I mean. That's why
Well,
like I don't want to be
you
like
have to go to the accessibility office and make your case there.
okay. All right. Thank you.
Okay. Okay. Okay, any other question? Okay. Bye-bye, everybody.